Hello, my name is Joyce and I'm an undergraduate in my senior year in the lab of Professor Gomez-Bombarelli in the MIT Department of Material Science and Engineering. Today, I'll be talking about our work on interpretable graph representation learning for lectin-glycan binding. This work is a collaboration between the labs of Professor Gomez-Bombarelli in the Department of Material Science and Engineering at MIT and the lab of Professor Laura Kiessling in the Department of Chemistry at MIT. It includes myself, Somesh Mohapatra, Omar Santiago-Reyes, Dr. Daria Kim, Professor Laura Kiessling, and Professor Rafael Gomez-Bombarelli. I'm very grateful for all of these collaborators for their contributions to this project. First, I will provide some background and explain the motivations for this project. Glycans are carbohydrate-based polymers made by all living organisms that play a major structural, energy storage, and regulatory roles in organisms. Glycans range from oligosaccharides that are only a few monosaccharide units long to much longer polysaccharides with a wide range of architectures that can range from linear to highly branched. Uncovering fundamental insights into the role of glycans in physiological processes, such as human immunity, however, requires an understanding of the interactions of glycans with glycan-binding proteins called lectins. Lectins are a class of proteins that bind to carbohydrates. Glycans at cell surfaces mediate the discrimination of human cells from invading pathogens through interaction with lectins, and therefore a solid understanding of lectin-glycan interactions remains essential for the development of novel diagnostics, therapeutics, vaccines, and beyond. A major tool for collecting data on lectin-glycan interactions is the glycan microarray, which consists of a variety of carbohydrates deposited onto distinct locations on a glass surface linked to the substrate via spacers. When a sample of a lectin is introduced, the binding strengths of several hundred carbohydrates with the lectin and relative fluorescence units, or RFUs, can be measured, and this produces a glycan binding profile for that protein. There have been several prior attempts to study glycan microarrays and interactions. In SweetTalk, developed by the lab of Jim Collins at MIT, glycans were encoded as glycowords composed of polysaccharide units. And this framework was used to classify glycans as immunogenic or non-immunogenic, as well as identify important motifs for glycan immunogenicity and pathogenicity. GlyNet, developed by the Dendritic group at the University of Alberta, represented an improvement from SweetTalk by encoding glycans as subgraph features consisting of monomers, dimers, and trimers. They developed a regression model that predicts relative binding strengths between mammalian glycans and glycan-binding proteins. Finally, Carl, developed by the Ramsden Lab at the Royal Melbourne Institute of Technology, takes a different but rather common frequent subtree mining approach to find motifs with a high frequency among positive binding glycans and a low frequency among negative binding glycans. They use this information to develop a classification pipeline to predict binding. So overall, although all of these prior attempts and approaches to study lectin-glycan interactions have predictive power, they lack the ability to reveal important monomer motifs and substructures, particularly substructures more precise than the individual monomer unit. We recently developed GLAMR, which is a framework for chemistry-informed graph representation, similarity computation, and interpretable supervised learning for macromolecules. Monomers were represented as nodes and bonds as edges, both featurized using circular fingerprints to capture the macromolecule's local chemistry and global topology. We obtained state-of-the-art results for the classification of glycans using graph neural networks, or GNNs. Using pre-trained supervised models for glycan human immunogenicity, we also demonstrated graph attribution to identify features, specifically glycan monomers and substructures, most critical for model decision-making. The glycan features and substructures that we identified that were critical for glycan human immunogenicity closely aligned with important motifs contributing to immunogenicity reported in the literature. For the first time, This chemistry-informed macromolecule graph representation provides a single framework for representation and interpretable supervised learning of natural and synthetic, linear and nonlinear macromolecules. I will now discuss the prediction of lectin-glycan binding strength and specificity using the GLAMR interpretable supervised learning framework. We used mammalian microarray data from the Consortium of Functional Glycomics for the lectins Concanavalin A, or ConA, and dendritic cell-specific ICAM-grabbing non-integrin, or DC sign. Each data point in the dataset represents a unique glycan and its corresponding binding strength to either ConA or DC sign in relative fluorescent units, or RFUs. On a log scale, the RFU values for ConA and DC sign follow a nearly Gaussian distribution, with ConA dataset having a slightly more bimodal distribution, as shown by the greater proportion of very strong binders. In order to better understand the problem we have at hand of predicting glycan-lectin binding strength, we performed an analysis using the GLAMR dissimilarity computation framework to analyze the relationship between the dissimilarity score to a reference glycan and the binding strength in RFU to DC sign across the entire dataset. We used a random glycan with high RFU or high binding strength to DC sign as the reference glycan, with which the dissimilarity score for all the other glycans in the dataset were computed. We obtained a Gaussian distribution for similarity, indicating high glycan-lectin specificity that cannot be uncovered using dissimilarity computation alone. Therefore, we trained a series of graph neural network regression models over minimization of root mean square error to predict glycan-lectin binding strength. We experimented with a wide range of input graph features, including circular fingerprints, the length of the spacer that links the glycan to the substrate, and 29 different physical chemical properties. In addition, the dataset provides 6 different interaction strength values in RFU for each glycan, corresponding with 6 different spots for the same glycan on the microarray. So we therefore tried including all of this replicate data in their analysis. In addition to different inputs and input features, we tried various output transformations, such as log transformation. We benchmark against many different combinations of these input features and output transformations to ultimately determine the most optimal combination for both ConA and DC sign. Our dataset for each lectin consists of 3,364 total points, and we trained on 60% of this dataset, validated on 20%, and tested on 20%. To determine the final validation and test metrics, we averaged over 25 total model implementations, the top 5 hyperparameter sets with 5 random seeds. On held out datasets for ConA and DC sign, the model ensembles had root mean square errors of 0.502 and 0.782 of the standard deviation of the training dataset, respectively. Our models for both ConA and DC sign outperformed existing models reported in the literature, demonstrating the robustness of our supervised learning approach. Not only were we able to predict lectin-glycan binding strength with state-of-the-art accuracy, but we also uncovered critical glycan substructures for lectin-glycan specificity. We performed 2 types of n-gram analyses to determine the most prevalent glycan monomers, dimers, and trimers in glycans with the highest binding affinity or RFU values to DC sign. We employed 2 different approaches, one simply using a weighted average of all RFU values of glycans containing each monomer, dimer, or trimer, and the other using a Mann-Whitney U-test. The results from the 2 methods closely agree with each other. And the tables on the slide show a list of top monomer, dimer, and trimer sugars found across glycans, sorted according to their contribution to RFU based on the first method that we used. The top monomers are fucose and mannose. The top dimer consists of N-acetylgalactosamine and N-acetylglucosamine 6-sulfate. And the top trimer consists of fucose, N-acetylglucosamine, and mannose. Graph attribution using the pre-trained GNNs helped to further decipher opaque structure property relationships providing significant information in the absence of ground truth understanding. For a randomly selected glycan that binds strongly to DC sign, we found that the monomers N-acetylglucosamine and fucose contributed most to binding specificity. In addition to individual monomers, critical substructures more precise than the monomer level were also uncovered using attribution. The highest weighted substructures within both highlighted monomers of the random glycan contain a cis-bisenyl hydroxyl group, which is a characteristic feature present in the binding motifs of sugar ligands that interact with lectins like DC sign, as reported in the literature. So in summary, we predicted lectin-glycan binding strength with state-of-the-art accuracy and uncovered motifs critical to the model's prediction of binding strength using an interpretable supervised learning framework. Supervised learning with GNNs provides a generalized system to learn over diverse glycan structures and lectin identities. N-gram analysis and graph attribution extract important sugar and substructures for specificity, with graph attribution for the first time revealing both key monomers and substructures more precise than the individual monomer level. This work therefore illustrates a robust method for elucidating the structural features responsible for critical biological processes such as lectin-glycan binding, providing a basis for future experimental validation and design of novel macromolecules. In the future, we will aim to extend this approach to include multiple lectins and generate new glycans for improved binding strength and specificity. Thank you so much for listening, and please let us know of any comments or suggestions either in the comments section of this video or live during the event. Thank you.